Yeah, thanks, Shaon, for your kind introduction. So, first, we are going to start with the pedagogical topic where I will be covering phylogenetics mostly. And then in the research talk, I will be talking about how to utilize the concepts in this field of phylogenetics to look at tumor evolution. Okay. So again, as Shaon mentioned, that my background has been in computer science. So today's both of these talks, I will be mostly focusing on algorithmic approaches for phylogenetic inference. And in the pedagogical topic, where I'm going to basically cover a set of different approaches for phylogenetic inference methods. And probably some of this might become more relevant when I start talking about tumor evolution in particular. But some of these, I would say, very basic concepts and approaches, they might be applicable to some other kind of data sets as well. For example, we'll be looking at some distance-based approaches of building phylogenies. And there you will see that even with other kind of data sets, which are not necessarily, let's say, optimal for phylogenetic inference, but there also you might encounter data sets using, for which distance-based approaches can be fruitful. Okay. So again, as of the first half of the talk, that is this 1.5 hours, I am going to spend mostly focusing on phylogenetics. This has not exactly anything to do with tumor per se, but these are more, I would say, basic and algorithmic approaches for phylogenetic inference. Okay. So with that, I'll start, or the story of this particular talk actually starts back in the era of Darwin. Okay, so who really came up with this tree of life, as you can see over here, and this was his hypothesis that basically that affinities of all the beings of the same class have sometimes been represented by a great tree. So he was looking at the biodiversity that is present across different ecosystems. And what he came up with the hypothesis that essentially all these different organisms that we see in today's world, they can be somehow connected via a very large tree. And this particular tree, what he actually called that this is the tree of life. So essentially, this is built upon these two particular principles that all organisms on Earth, they really have a common ancestor. And that's how, through this tree structure, any set of species are actually related to each other. So these are sort of the basic principles based on which phylogenetics is built upon. Okay. So in fact, this research actually was carried forward by other biologists as well. So there was a German zoologist, Ernst Haeckel, who really coined this term phylogenetics, okay, or phylogeny. And in fact, here I am showing you some of these trees which were actually constructed by this person. And what you see is basically here again, we are looking at again tree of life, where the species, they are basically categorized into three different groups, plants, protists, and animals. And here is another very famous, I would say, tree of life, which is really the pedigree of man. Okay. So what you see here is at the leaf of this particular tree, which is rooted over here. At the trees, you see all the different species, and then the tree structure is actually talking about the evolutionary relationship between the different species. Okay. And this is where we are looking at a much more modern version of this tree of life. And this is also built upon molecular data. So that means genomic data sets, sequencing data sets were utilized for building this tree of life. Okay. And what you see over here, these different colors are also representing a large class of species in, which are very much related to each other. So for example, eukaryotes are over here, okay? And then you have a lot of different bacteria, archaea, and all these organisms. So these are actually all bacterial species, and there are archaeal species over here, and so on. And this again, this is representative of the tree of life for a large number of species. Now the question is, once we are given this kind of molecular data sets, this kind of genomic data sets, how do we really go about building this kind of tree? Okay, so this is just one such example. And this is another example which became very relevant, I think, during the pandemic time. People were dynamically screening a lot of different, I would say, samples, clinical samples, and they were trying to sequence this virus, really different strains of the viruses, and they were looking at how things are actually changing dynamically, right? So this is at some particular time where you really see that the ending time point is kind of like the December 2022, and you see a lot of different, I would say, variants of SARS-CoV-2, including the different Omicrons, Delta variants, and so on. Okay, so with that, with this brief introduction, let's actually define phylogenetic tree. How to really define phylogenetic tree? Okay, so essentially, this is the underlying principle that when there is a group of aligned sequences and they are showing significant similarity to each other. So when I'm saying aligned sequences, I am actually talking about an output of a multiple sequence alignment algorithm that you really start with a set of DNA sequences, or let's say a set of protein sequences, and you have performed multiple sequence alignment using one of the existing algorithms, okay? And once you see that this set of sequences, they actually show some similarity to each other, that means there are certain regions which are conserved across all the different sequences, then basically it can be taken as evidence that they are somehow related during the evolution, okay? And essentially they came from a common ancestral sequences as the result of this divergent evolution process, okay? And all we are trying to do, we are trying to reconstruct this evolutionary history, okay? And the way we actually reconstruct this evolutionary history is that we want to represent them as a graphical structure, and this graphical structure is sometimes called a phylogenetic tree. And this is essentially the definition of phylogenetic tree, that we have a set of sequences which are connected via a graphical structure, and this graphical structure is nothing but a tree structure also. And when it is a tree structure, then we are actually calling it a phylogenetic tree. Now, in order to build such a phylogenetic tree, we need a set of sequences, okay? And this set of sequences should have a common ancestor and common function. And this kind of sequences are sometimes called orthologs, okay? They can be coming from different species, okay? And we can build this kind of phylogenetic tree based on, let's say, a whole genome sequencing data set, like where you are basically looking at sequences from the whole genome at the whole genome level, or you can also look at it from, let's say, one gene at a time. And depending on what kind of data you are using, it gives rise to different types of tree structures. For example, if we are looking at the evolution of species, then we call it a species tree. Or if we are looking at the evolution of only, let's say, as a single gene, then we call it a gene tree. And then sometimes there is incongruence between this kind of gene trees and species trees, because the evolution of the species itself can be somewhat different from evolution of a single gene. Again, we are not going to complicate, I think, our talk by looking into this kind of incongruences, but for the time being, let us just focus on the underlying principles of, given such kind of data set, that is a set of aligned sequences, how we can really build this tree structure. Because then we can make a lot of hypotheses that what was the common ancestor given a set of sequences, what kind of divergence was actually responsible for this speciation event, and so on, right? So with that, again, let us talk about some terminologies. So for example, when we are talking about phylogenetic tree, we are talking about a 2D graphical structure, as you are going to see some structure in later slides. And then at the leaf of these trees, we have something called taxon, okay, or operational taxonomic units. So what are exactly this? So essentially, if this is a very simple tree, okay, this is the root of the tree, this is drawn upside down, and these are basically leaves. So you have L1, L2, L3, L4. So this L1, L2, L2, L3, L4, These are known as taxon or OTU. Okay, what are these? These are the objects basically on which we are building this tree. Now you can imagine that by having different placements of L1, L2, L3, L4, we will end up with a different tree structure, really. And this tree structure over here, let us denote it by T. So this is what we are calling as a phylogenetic tree. Okay. So now we are going to look at more definition of this phylogenetic tree very soon. But for now, you have to remember that in order to build any phylogenetic tree, we need a set of taxon. So these are basically represented when we really want to associate this set of leaves with the set of data, then each one of these leaves are actually corresponding to one of the DNA sequences, let's say. Okay. So this is what illustration of a phylogenetic tree actually looks like. So what you have over here is one type of representation, and on this side you have another type of representation. So what is the similarities between these two representations and what are the differences? So what you see here, all these different birds, let's say these are imaginary set of birds which are representing different species. And what we can see is that some of these are actually representing some internal nodes, some of these are representing leaves, and there are branches which are actually connecting these different birds species. Okay. So there are certain things to look at. First of all, one of the property that we see that these are binary tree. What are binary tree? So if you take a look at any of these internal nodes, okay, in the tree, you see that they are connected to 3 other nodes, okay. And if you look at these external nodes or the leaves, they are connected to only one other node. So this is really the definition of a binary species tree or binary tree in that sense, that any internal node will be connected to 3 other different nodes, and the external nodes or the leaves, they are connected to only one other node. Okay. What you see over here is another thing, which is that this particular structure doesn't seem to have a root. Okay. So that's why it is called unrooted binary species tree. In comparison or in contrast, when you are seeing over here, here you see that one particular internal node is defined as the root of the whole tree. So what is happening is that we have these different branches, but we also have this root of this whole tree structure, and any branch where there is a bifurcation happening, this is denoting a speciation event. When we have defined this root also, then this is also indicating a direction of descent, that is the flow of time. So at the root itself, that is like the very initial time, and as you move down the root to any of the leaves, then basically you are going forward in time. Okay. So that is what this rooted binary tree actually represents. So that means in order to represent a phylogenetic tree, we have to have idea about at least this set of things. That is, we have a bunch of leaves, which are basically OTUs or taxons, right? On which we are building the tree ultimately. But at the same time, we have this set of internal nodes, which are actually representing ancestral states, okay? So that means either some of these species or these internal nodes, either they are representing certain species which are hypothesized to have occurred during evolutionary process, okay, and we are not seeing them at current time point from where the snapshot or the data has been taken. The data has been taken only from these leaves, right? So these are the set of observed species. Now some of these may be extinct as well, or in most cases what we say is that they are actually existing species. But in some cases, in some particular examples, you may have particular species which have really become extinct, but maybe you have some traces of the DNA or anything, okay? And ultimately, these branches that you are seeing, these branches are connecting different nodes, and these branches are actually corresponding to the evolutionary relationship between the different species. So if you have all these different things, that is nodes, again, internal nodes as well as external nodes and the branches, then you have the overall phylogenetic tree structure. Yes. Yeah. So, mhm. Yeah, very good question. So regarding the length, you will see later, I will be talking about a specific, I would say, algorithm to define the length or determine the length of the branches. But if you think about determining the root, right, it actually sometimes you have to apply this kind of domain knowledge. Let's say you are looking at a set of bacterial species, okay, and you know that you have 100 bacterial species which are somewhat related to each other. Then what you do, you take a very different bacterial species or something very different, okay? So this is sometimes called an outgroup. You know that this particular species is very, very different from all the other sequences. So then you can define a root which is basically separating this outgroup with the rest of the sequences. So sometimes we have to do this kind of, build this kind of data sets to sort of define the root. In some other cases, like as you will see in case of cancer also, we know that we can define probably a normal cell as a root, okay? Or if we have a bunch of normal cells without containing the somatic mutations, then we can say that there is a root which is separating all these normal cells against all the cancer cells. So that is how you define the roots, okay? The- So what you are actually referring to is again binary tree as used in the data structure, right? So again, the data becomes completely different. Here you are looking at biological sequences. So you have to define the root in that manner rather than going into the computer science constructs. Okay.
Again, one more question. So, just like in decision trees right now, since people have related to computer science, and why not to relate it with machine learning as well?
Yeah.
But I wanted to ask, so why decision trees where we extract certain features, right? And this could be the cell wall, right? Could be presence of mitochondria, presence of circular DNA, and so on and so forth. Based on these features, we could again construct decision trees, and we could select which decision tree best represents the phylogenetic tree. Does that make sense, or like kind of, because I see that phylogenetic tree also has a lot in common with decision trees, because just the thing is that we add the domain knowledge there.
Well, I would say that when you are talking about decision trees, again, you are choosing a node to really split the data, right? And you are using some sort of principles such as information gain or Gini index to do it. And here, whenever we are talking about features, we are really talking about how much amount of divergence you see in the sequence level, at the sequence level. So I would say one can still probably apply some sort of, I don't know, entropy based formulation to sort of select, okay, should I split the data at the very beginning or maybe later? It is somewhat similar. You will see again, it is really dependent on the presence or absence of certain markers, really. But I would still say probably the construction of this kind of trees will be somewhat different from that of decision tree. Okay. Okay, so now coming back to again some terminologies, simple terminologies. We have this binary tree or bifurcating tree. So this is the definition I already mentioned, that every internal node should be of degree 3, that is, it connects to 3 other different nodes. And then every leaf is of degree 1, that is, it connects to only one other node. Okay. Now, is this the only kind of tree that is making sense? No, not necessarily true. There can be multifurcating trees also, where some internal nodes can have higher degree. So why that can happen? Again, we are going to see some examples later, like why that can happen, why you can have multifurcating trees. Okay. Now, this is actually a simple question to all of you. Maybe you can think about it and let me know. If you have a binary tree which is built upon n taxa, okay, then in that particular tree, how many nodes do you have, total number of nodes, how many branches do you have, and how many internal nodes and internal branches do you have, okay? So we'll come back to this question later. So now, as I mentioned that there are two components in representing a phylogenetic tree. We have this tree topology, that is the overall branching structure, okay, the graphical structure itself, and then the branch lengths. Coming back to one of the previous questions, that how to determine the branch lengths, we'll come back to that. But these two things together actually represent the phylogenetic tree. Now, in some cases, or particular to some applications, maybe the tree topology becomes more and more important, but in some other cases, branch lengths can also be some important parameters to learn, okay? So now, just to give you sort of the number of possibilities. So in the first column, what I am showing you is the number of taxa. And the second column shows the number of possible unrooted trees. And the third column shows the number of possible rooted trees. And you can see how quickly these numbers actually grow. So if you have 10 taxa, okay, we'll start from something simpler. If you have 4 taxa, you can only have 3 possible unrooted tree structures and 15 possible rooted tree structures. But as soon as you go into something like 10, you see these numbers are already in millions, okay? And this is basically the actual mathematical formulation giving you how quickly these numbers are growing. So these are all growing factorially. So you can imagine that if we really want to study, let's say, the evolution of something like 20 taxa or 30 taxa, it's already giving us a number of possibilities which we probably will never be able to exhaustively search in practice, okay? So that's where the challenge really comes from. So let us actually take a look at different possibility of rooted trees given an unrooted tree. So here is an example. You have 3 taxa, 1, 2, 3, and now you can possibly think about how to convert it into a rooted tree, okay? So it turns out that there are 3 possibilities to convert it into a rooted tree. And all these 3 topologies are actually different from each other. What is happening in the first case, it is saying that 1 is very different from 2 or 3. So the root is being placed on this particular branch, okay? In the second case, root is being placed on this particular branch, which is connected to the species number 2. And here, as a result, what is happening is that 1 and 3 are similar to each other, and they are slightly distinct from the species number 2. And then we have this third possibility where we are placing the mutation or the root on this particular branch, and we see, okay, 3 is actually different from 1 and 2. So just corresponding to a single unrooted tree consisting of 3 species, now you have 3 different possibilities of rooted tree topologies, okay? So can anyone tell me how many rooted trees are possible for this particular unrooted tree? Anyone? Yes, and how are you saying it's 7? N minus 1. What is n? Number of leaves? Okay. And what is the number of leaves in this case? So certainly n minus 1 is not the answer, right? So it's basically the total number of branches that you have on this tree, because you can place the root on any of these branches, and corresponding to that you will end up with a different rooted tree, okay? So now coming back to this question of bifurcation versus multifurcation. So what we are seeing over here, okay, this is a specific type of tree which is also called as a star tree. What is this? You have a single root, and all the other, I would say, nodes or these leaves are all connected to this particular root. So what we are saying that this is an unresolved tree topology from which we have no idea whether any of these leaves are more similar or distinct from each other or not, okay? So that's why it is called completely unresolved tree, And the presence of such a node is also known as polytomy or multifurcation. Okay, so essentially, if you have any internal node which is connected to more than two nodes, okay, then it is a polytomy. It is an example of a multifurcation or polytomy. So let us see at the next example where we have we can see that B and D, at least their lineage history is kind of resolved, but there is still some amount of polytomy or multifurcation remaining. A, C, E, between these three, we cannot choose two particular species or two leaves. We cannot say that A and C are more closer to each other as compared to E. Okay, so this is still partially unresolved phylogeny, because for part of the tree, the lineage history is completely resolved, but for another part of the tree, the lineage history is not resolved. Okay, and then we have this fully resolved bifurcating phylogeny. So anywhere you see all the branching that you see over here, these are all bifurcations. So that means this is fully resolved, and from this tree structure, we can clearly say which species are more similar to each other and what are the different common ancestors for different set of species. Okay. So essentially, if we are moving from completely unresolved tree towards fully resolved tree, we are increasing the resolution definitely, and we are removing this kind of polytomies. Now, why this kind of polytomies can actually occur in practice? So let's say you have a bunch of sequences for which it's very difficult to determine whether, let's say this scenario, right? It's very difficult to say that A and C are closer to each other as compared to E, or let's say C or E are closer to each other as compared to A, because you do not have probably enough signal in the data. Okay, so at the best we can say that all these three species are related to each other and they are very different from all the other set of species, but we cannot really determine an order during the evolutionary process, the order they really followed. Okay, in the real evolutionary history, you probably will not have something like a multifurcation, because the speciation events are assumed to be somewhat binary in practice. But then again, there are a lot of evolutionary events which can complicate this kind of things as well. Like there are events like incomplete lineage sorting or horizontal gene transfer, which can make things even more complicated. But if we just look at the speciation particularly, then we can say that ultimately at the level of the molecular evolution, yes, the tree should be actually looking like a fully bifurcating tree, okay, because we are looking at molecular changes that are really happening at the DNA or the protein level, right? But maybe because of the lack of the data, we have to resolve in this manner, in where we are ending up with this kind of multifurcations in the tree. Okay. Yes. It is possible. Again, what you are talking about is basically, again, going back to the same system. Here I am talking about more like speciation or change in the molecular sequences itself. So when we are talking about molecular changes, then we are talking about that history should be bifurcating, and maybe we do not have enough data to resolve that. That's when we end up with polytomy. Convergent evolution. Convergent evolution is something very different, right? Where convergent evolution says that you have similar functionality being developed in, let's say, two different organisms, but there is no real molecular mechanism involved. Okay, that is convergent evolution. So, for example, let's say wings in bats or wings in birds, their actual evolutionary history is different, but they are functioning similarly, right? So this is an example of a convergent evolution. And we see some convergent evolution even at the molecular level also. So for example, what we see is that, let's say we talk about differentiating systems, right? Cells differentiating over a dynamic process. And in some cases, the same cell types actually can arise in two very different lineages. The same cell types, because they have the same set of functions, or their gene expressions are very, very similar, right? but their lineage history is very different. So that is also another example of convergent evolution that we see. Okay, so now we talk about another type of terminologies distinguishing different types of tree. So what you are seeing here is different types of tree. Again, these are types of tree using which we can represent the data sets. The first set is very, very simple. It is called as rooted cladogram. Okay, what is it showing? Here, you are just looking at the structure itself. The branch lengths, they have no meaning. Okay, and basically these ancestors are implied by these internal nodes. So what is meaningful from this kind of tree is that you only have the topology of the tree. You can only say that, okay, these two species are more closer to each other, these two species are more closer to each other, but really you cannot put any numbers on the branches. Okay. The second type of tree is sometimes called as additive trees. So here branch lengths are actually denoting some measure of the evolution. So what you see here is that you can see that the branches are of different lengths, and there are numbers associated with these branches. So essentially these branch lengths are actually proportional to the number of mutations happening per site. Okay, so by this principle, going by this principle, we can say that this particular species is actually acquiring a lot of mutations as compared to, let's say, some of the recent common ancestors and so on. And we can also say that, okay, the difference between the mutational difference between these two species are much less as compared to these two species, right? Just by looking at these branch lengths. And the reason it is called as additive trees is because if you sum up the distances by traversing the paths in the tree, then you can actually say how much is the mutational divergence between these two species. So that's why it is known as additive tree. So there is a third type of tree which is called ultrametric tree. And this ultrametric tree is basically assuming a specific biological property which is known as molecular clock. So it's an assumption, it's a modeling assumption. It assumes that the mutation rate is constant across all the different branches of the phylogenetic tree. Okay. So what is it basically saying? It is basically saying is that when you start from the root and you traverse any of these paths across, let's say by taking any of these bifurcations, and you reach a particular leaf, okay? So then if you are summing up all these branch lengths, let's say you start from this root, you are taking this branch, again this branch and this branch, so you are coming to this particular species over here or the leaf over here. In contrast, let's say you are taking this path and you are reaching over here. The distance that you traverse from the root to the leaf is going to be the same. Why? Because essentially it is assuming that mutation rate on any of these branches is constant, it's the same. Okay. So this is the molecular clock property, and it is useful in terms of modeling some data sets, and you will see the use of it when we talk about a specific method for reconstructing phylogeny. Yes. Not necessarily true. You can just look at any site. Okay. Of course, this is actually a very simplifying assumption. You can say that this is probably not true, biologically speaking. But sometimes we have to make this kind of assumption for the practical purposes of analyzing the data sets. And even by utilizing, let's say, this kind of molecular clock property, we can end up having some, I would say, we can still derive at some hypothesis about the evolutionary relationships between the different species. Okay.
Hamim, so
Yes.
if in ultrametric as well as in the additive types, I was just trying to relate the two. Number of mutations is all that counts to segregate the, to perform a speciation event, right? Mm-hmm. So if in ultrametric, the length of the line also represents the number of mutations that have occurred, because the larger, because the rate is constant of the number of mutations. So essentially what would segregate is how much mutations do, cells or any speciation event
So what you can
accumulates.
see again, if you have to compare these two trees, right? One fundamental change that you see over here, that from the root to the leaf, the distance is remaining constant in this case, just because of this constant mutation rate property. But here that is not true. If you take from the root to any of the leaf, you count the distance, it is actually variable. Okay, and that is where the difference comes from. So this is actually a more restrictive assumption, saying that you have to assume a constant mutation rate, which may not be biologically very, very plausible all the time. But this is actually a more generalized scenario where you are really, your branch length is saying, of course, here also your branch length is saying some measure of evolution, but with the added constraint that from the root to the leaf, the distance is going to be constant. But that is not the case over here. Okay, so that is the major difference, I would say. So this is, in practice, this is more useful. Yes. Can you see say that yellow bird with 6? Yes. So would that have more mutations than the brown with 4 on that branch? So that we can say. Yeah, that we can say. Exactly, that is precisely what we can say. Is that this is more distant from the root as compared to, let's say, this one. Just because it is acquiring more and more mutations. Okay. It is still more similar to this one, because at least they are sharing certain mutations which are actually common. Again, when we talk about, let's say, reconstruction, then you will see some of this. Okay, and this is just an example, like how you root a particular tree, that you have really added this orange bird, which is very distantly related, so it is not related to any of these other birds, and that is how basically this is an outgroup through which you have rooted this additive tree. Okay. So I think I am going too slow, so I'll try to, again, we'll see how much we can really cover, but maybe the more important things I'll try to cover. So as you saw, the phylogenetic tree structure and everything is very good visual structure. It's good for our human interpretation. But when you really have to make the computer understand, okay, how the tree structure is changing, let's say, or what is the tree structure, then how do you represent this kind of tree? Okay, so the way you represent a phylogenetic tree is in terms of splits or bipartitions. Okay, what is it? Basically, a split is going to divide the set of taxa into two different, or into collection of subgroups, let's say. Okay, so what you see is that every branch in the tree, they are connecting two sets of nodes. So if you remove that branch, you will end up with one set of nodes on one side, another set of node on the other side of the tree. Okay, and each tree is basically a connection of such branches, collection of such branches. That means when we are talking about these nodes, the set of nodes, each branch basically divides the whole set of nodes into two subgroups, right? So that means that is like a split or a bipartition. So if you take, for example, this is a tree. If I want to split this particular branch, or if we just remove this branch, then one side we get this set of leaves or this set of nodes C, D, E, F. On the other side we get A and B. So this is basically saying that this is a bipartition, and through this bipartition, this is how the nodes are actually subdivided. Another bipartition could be somewhere here, and you can say that, okay, on one side of this branch we have ACB, on the other side we have DEF. Okay, so essentially a tree is containing as many splits as there are branches. Of course, there are some trivial splits which are not telling anything about the tree structure per se. What are the trivial splits? Any branch that is connected to a leaf, if you cut the tree there, you will end up with, on one side you will have, let's say, this one, A. So this is one set. On the other side you have everything else, B, C, D, E, F. So any branch which is connected to a leaf gives you a trivial bipartition. it's the non-trivial bipartitions or the splits which really talk about the tree structure. Okay. So this is an example of a non-trivial bipartition, and this is an example of a trivial bipartition, really. So in this particular tree, if you count the total number of bipartitions, there will be total 9 bipartitions. 3 of them are non-trivial. Which are the non-trivial one? This is the non-trivial, this is another non-trivial, this is another non-trivial bipartitions. Okay. And similarly, there is a concept of clade in rooted tree. So what is defined as a clade? This is a group of leaves that have a common ancestor, okay? And that is not a common ancestor for any other leaf in the tree. That is the definition of a clade. So if you look at this rooted tree, human, chicken and Xenopus, they are forming a clade. Because why? There is a common ancestor. So this is the common ancestor, this is the common ancestor for all this set of leaves, okay? And this particular common ancestor cannot be a common ancestor for any other leaf in the tree. So this is the example of a clade, and if you count the number of clades over here, you will have 11 clades. 4 of them are non-trivial. Why? Again, what are the trivial clades? Any sequence or any leaf itself is a clade, right? That is by definition. And then the overall tree is also another clade, right? Where basically root is the common ancestor of all the sequences. So after discounting them, you will end up with 4 non-trivial, I would say, clade in this particular example. And the reason for talking about all these splits or bipartitions or clades is because we want to somehow represent the tree structure using some sort of, I would say, representation that is better for the computer to understand. And one such format is called Newick format or New Hampshire format. So essentially what we are doing is we are writing each of the splits in a bracketed list of the taxa. Let's take an example. I mean, let's try to define how exactly to convert this thing into Newick format. Okay. So this is the Newick format of this whole tree. So this is exactly saying the tree structure, or this is exactly encoding the tree structure as you can see in this diagram. How that is the case, okay? So it says that each split is written as a bracketed list of the taxa, okay? So let us start from the beginning. What we see here, bear and raccoon, okay? They are actually forming a clade, right? So that is over here. Bear and raccoon together, they are forming a clade, so we are writing inside parenthesis. Then we see bear, raccoon and dog, they are also forming another clade. Okay, so this is basically a split. Bear, raccoon and dog, they are on one side of the split, everything else is on the other side of the split. So that is basically represented over here, dog, bear and raccoon. Okay, so if we go on like this, then we can see that everything will be represented like this. Okay, so in the end we have till monkey, and essentially it's saying that the root is basically connected to monkey on one side and everything else is on the other side, and then this is recursively defining the overall tree structure. And in the end what we have this semicolon, which is basically denoting the end of the tree. This is where the tree has ended. So this is a string which is representing the whole tree structure, how the different splits are present in this particular tree structure. Okay, and this is called the Newick tree or New Hampshire format of the tree. Now, is it the case that we can only associate the tree topology, or we can only encode the tree topology using this string representation? No, we can actually be very general. We can add more information. For example, we can add this kind of information such as branch length. This can be added. So just to give you a particular example, let's say we have written that dog : 0.2. It is basically saying that the branch that is connecting dog to the common ancestor has length 0.2 and so on. Okay. So using this Newick format, you can even represent branch length, You can represent even some other kind of information. Let's say you want to have certain other annotations on the branches. You can also represent that using Newick format.
If it's a symmetric tree, then like if the same thing would have happened on the downside, right? So there is bear raccoon, very similar relation. If it's a symmetric tree around this monkey line.
What do you actually mean by symmetric
So
tree?
I mean that there are more such clades, and the pattern of clades that you see on top, and the same thing would have happened at the bottom. Does that make sense?
No,
doesn't.
nothing like that. This is the overall tree structure, right? So I think what you are trying to suggest that whether the same structure is repeated on this bottom side. No, that is not true. This is just the tree structure, and corresponding Newick format is just this. And we are just representing the different splits using this parenthesis. That's all we are doing. And this is for basically, you can use this string format, you can give it to the computer program, and then it will try to, basically there are programs that will build this tree structure for you. Or you can write your own data structure as well, essentially. So I think for the sake of time, I am going to move over this part where we are talking about consensus or condensed trees. This might be, if time permits, then we'll come back to this. But now I will move on to how to really reconstruct this kind of phylogenetic trees. Okay. So before that, if there is any question, please feel free to ask. Okay. If not, then I think we'll get started with the approaches for phylogenetic tree reconstruction. So we really need to understand or answer this kind of 4 questions. The first question is, what kind of data should we really use? The second question is, which method we should be using? The third question is, what kind of evolutionary model? Because ultimately we are saying we are trying to model evolution, right? That means we have to have a model of evolution. It can be simple, it can be intermediate, it can be complex, depends on what you really want to get from the model. And finally, once you have reconstructed everything, is there any particular test that we can perform in order to test the robustness of the prediction from the tree structures? Okay, so these are the 4 things that are very important. So the first thing is the data. And as we mentioned that we should be working with data which is basically coming from orthologous sequences from the different species. So that means we are trying to reconstruct this evolutionary history. That means we should utilize the sequences which has the traces of evolution. Okay. So essentially, again, it comes down to the, I would say it varies from one problem to another. We are working with, let's say, something like a species tree or gene tree that we want to build. Then we should be working with orthologous sequences. And then this particular property is very important, that the rate of change in that region from which we have taken the data, right? It should be fast enough to distinguish between closely related species, but then not too fast that regions from very distant related species cannot be reliably aligned. What do I really mean by that? What I mean by that is that the sequence, the piece of sequence from which we are going to build the tree, they should have some region which are highly conserved. So that is really good for distantly related species. Let's say we have a specific region which might be something like a transcription factor binding site, right? And essentially which is conserved across a lot of different species. So they are the highly conserved regions. On the other side, there might be regions which are more variable between the species, and this is really good for closely related species. So if you have, let's say, one group of bacteria which are very similar to each other, if you have no distinction across all these different bacterial species, then probably you will not be able to resolve their phylogenetic history, and then you will end up with a polytomy, right? But at the same time, you cannot have completely variable regions or completely variable, I would say, sequences all the time. Then essentially you are losing this kind of distantly relation with respect to this kind of distant related species. So if let's say in your particular data, there is no site which is conserved at all, then you cannot say anything about the evolutionary relationship with respect to this kind of distant species, right? So this is a particular thing to notice. And again, here we are talking about species evolution, but when it comes to cancer, let's say, then we are working with an evolutionary system which is not in millions of years, but rather, let's say, some particular years, 4 or 5 years time. Of course, with heightened mutation rate. And then we are left with sort of the positions on which we are actually observing this kind of mutations, right? Because those become our marker for distinguishing different cancer subpopulations, let's say. Yes. Yes. So I think what you are talking about is phenotypic changes. Here I am talking about something at the level of the genome, right? So even with, let's say, same genomic configuration, you can still have some sort of phenotypic changes. But here we are really talking about changes in the DNA, changes in the DNA level. Yeah, you can utilize epigenetic changes as well. As long as you are deriving the features out of that, it can be utilized.
But I mean, following that, then in epigenetic changes, if I thought that there shouldn't be possibility of mutation at the same site, right? So you know what I mean, that the reversion of the mutation event.
Yeah, all those things can be modeled using a
modeled.
system like this. Yes, you will see, like, as I am talking about the model of evolution, there you will see that there are different models of evolution. Some of them very simple, some of them more complex, and then it can be modeled. Yes,
Let me go
please.
back to the question that was asked. So, I mean, it seems like if you start talking about things other than genetic, or even if you talk about genetic and allowing reversion, then you're no longer talking about a tree, you're talking about something with roots, aren't you?
No, not
Well,
necessarily.
because now you go back, you can go backwards.
You can go backwards.
Make it a branch that then merges with a different branch because the distinguishing chain disappears.
So
it's not heritable
So when we are talking about, let's say, reversal, you will see, I will show some example, one particular example I'll show. You can have the same site mutated multiple times in the past, right? And then maybe what you are seeing at the present time is the current state of the genome, but there can be like 4 or 5 changes that actually happen during evolutionary time. You cannot really say or cannot really infer everything, but you can still model it that if my observed differences is something like this, if we say if we say that every site is getting mutated under a certain model, then according to that model, you can have some corrections introduced. And this loss or the reversal that you are talking about, particularly this becomes very useful in the context of cancer. I'll show you one example later.
Take multiple variables, right? It won't be dependent just on the single, I wanted to shift to new bivariate or multiple, you'll have to look at the correlations maybe in the clades, because maybe if this site has reversed its mutation, right, the other sites which are conserved and belonging to a particular clade, then you would infer that, probably this is a reversible mutation.
I think when I start talking about the model of evolution, it will be more clear. Okay, so let's just wait till that. So second thing is the choice of the methods, really. Okay, so over time, I think this field of phylogenetics, which is really a subfield of discrete maths, I should mention. So over time, lot of computational methods have been developed. Okay, and broadly they can be classified into 2 groups. First of is basically distance-based methods, and the second one is basically known as sequence-based methods. So what is the distinction? So if we talk about distance-based methods, essentially what they do they take the multiple sequence alignment data, they really compute pairwise distances from this set of data. And once these distances are computed, the multiple sequence alignment is thrown away. All we are working with is this kind of distances. And then these distances are basically converted or used for constructing the tree structure. What are some examples? These are some examples: UPGMA, neighbor joining, Fitch-Margoliouth. And UPGMA is basically the simplest algorithm for constructing phylogenetic tree, if I have to say the distance-based methods. And you will see UPGMA is very, very similar to hierarchical clustering. Okay. Neighbor joining is another method which is working under a particular evolutionary model, that is, and this particular method is also very popular because it's very fast and gives nearly accurate result in many cases. Okay. On the other side, we have this sequence-based method. So instead of computing some, I think, secondary information, they are actually working with these multiple alignments directly. Okay, so they are evaluating all the different sequences at each alignment site, and we'll see how they are doing so. And ultimately, what they are doing, they are searching the tree space using some sort of optimality criterion. Okay, what can be the optimality criterion? They can be parsimony based, likelihood based, or then we can have even Bayesian models of evaluating all these things. Excuse me. Okay, so we are going to see some example of each of these methods. So it varies from problem to problem. So for example, in some cases you can have 1,000 bases or even 10,000 bases. That is again usually in the I would say genes tree or species tree reconstruction. But when you were concerning, let's say, evolution of cancer cells, we are really using the marker positions, that in the mutation sites, which can vary. Let's say in a targeted sequencing case, you can have 50 sites. Whole exome sequencing can give you something around 1,000 and so on. So it varies from one problem to another. So this is actually just a table. I do not want to go to the details of this table, but this is showing the different features of the different methods that I just talked about, whether they are resulting in unrooted tree or additive tree or ultrametric tree, whether they result in a single tree topology or they can infer multiple trees and so on. Okay, and you see there is a bit of variability of how they are utilizing the available data sets and what kind of results they are inferring. And these are basically set of softwares that are commonly available and which are used by a lot of labs across the globe. Okay. Now, the third consideration, which is basically the choice of model of evolution. Okay. As I said that we have to particularly come up with a model of evolution. It can be, we can start from a very simple model. Okay. And then we can slowly build more and more complex model of evolution. Yes. So again, when I actually start talking about the details of the methods, it will be more clear. But you will see that distance-based methods, sometimes they are chosen to sort of get to an initial solution. Okay. So let's say you are working with something like likelihood-based approach. You will still apply first neighbor joining. You will get to a tree structure, and you start your maximum likelihood approach from that neighbor joining tree. So sometimes these distance-based approaches are used to get to a very good solution in the tree space, so that you can actually hover around that particular tree. And because these are very fast methods we are talking about. Whereas, let's say something like maximum likelihood or maximum parsimony, they are little slower as compared to distance-based method, but they are more robust in terms of the search. Okay, so again, it varies from one problem to another, but I think one good recipe is that you can start with this kind of distance-based methods, and once you get to a solution, you can start that start your sequence alignment based methods from that distance based tree. Yes.
Say ask all the questions I always wanted to ask. So, is the multiple alignment is done separately? There's never any iterative step where you go back and say this tree didn't work so well, and maybe my multiple alignment wasn't so good.
It is possible. In fact, there are set of multiple alignment methods which are using a guide tree. So iterative, I think there are iterative processes which can go back and forth. That you build a tree and you can say, okay, this tree is not making a lot of sense, so maybe I should change my alignment. Yes. Yes. Yeah. So as you will see that when you actually start with this kind of alignment based methods, you are getting something for free, which is basically a test or the, I would say once you obtain the solution, how robust is your solution, you can test that, which you cannot obtain or which you really cannot do with distance based methods. So after you obtain your solution, you can still run some more tests to say whether your solution is good or bad. Okay. Okay, now coming to this scenario of model of evolution, right? So we'll start with some very basic models. Again, I'll show you some basic model, and then one can go about making it more and more complex, because ultimately when we are trying to model this kind of data sets, we have to take into account what kind of events may have happened, right? So there is some amount of trade-off though. When you are actually starting with a very simple model, if your actual evolutionary history is more complex, then maybe your simpler model cannot do very well. But still, it will have some generalized idea, right, about your hypothesis. On the other side, you can have very extreme complex model, extremely complex model. They will be containing more and more parameters, and they can fit the data very well, but they will not be that much generalizable. So you have to do a trade-off between complexity of the model and generalizability of the model. And so you will see that the models even that I'm talking about, they are starting from the simpler model, and slightly more complex model is basically containing the simpler model, okay? Or the simpler model is basically a special case of the more complex model. That's what actually happens. So sometimes there is a concept of nested models, that one model is nested within the other model, and then you can perform some sort of statistical test to evaluate whether your data is more explainable under model 1 versus model 2. AIC and BIC, or there are hierarchical likelihood tests you can perform. Okay. So again, this is something that I was saying earlier, that you can start with something like distance-based method. They are very fast, and essentially after that you can apply this kind of sequence-based method. Now the question is, when this evolutionary model is being applied, you will see that in case of distance-based method, the model is basically used to convert the usual alignment data to the distance matrix, okay? And during the tree reconstruction, the evolutionary model is not being used further. Whereas in case of sequence-based method, the evolutionary model is being used at all stages of the tree reconstruction process. So just to give you some examples of this model of evolution, so I'll start with the very simplest simple model, which is known as Jukes Cantor. It is really from 1969. And what it is doing, it is just saying that the base composition of DNA is the same for the 4 different nucleotides. So they are present in 1:1:1:1 ratio. The transition and transversion rate is same, okay? They are not distinguishing between transition and transversion. And they are basically saying that there is a single rate or single parameter associated with any mutation. So let's say that parameter is alpha. And with that, we are going to model the system. So this is the most simple model. Then comes, let's say, these two models, Felsenstein 81 and Kimura 2 parameter model. So what is Kimura 2 parameter model doing? Everything is same except they are saying that transition and transversion rates should be different. Okay, so they are using two parameters, alpha and beta. And that is why Kimura 2 parameter is more generalized model as compared to Jukes Cantor, and Jukes Cantor is just a specialized scenario of Kimura 2 parameter. And these two models, we can say that these are nested models, examples of nested models. Similarly, Felsenstein 81, it is everything is same with respect to Jukes Cantor, except it is assuming that the base composition might be varying because, well, for different species, the amount of the nucleotide or the fraction of the nucleotides can actually vary in a sequence, right? So likewise, we have this, I would say, this generalized time reversible model. This is the most complex model. It is assuming everything that, okay, there is variable base composition, transition transversion rates are different, even every mutation rate is different. So it is consisting of more number of parameters. And then it basically becomes more complex, but then you can try to evaluate all these model parameters. And then you can try to run something like AIC or BIC to determine whether this explains your data better or as compared to something else. Okay. Yes. So let's say these two models you are comparing, right? Jukes Cantor and Kimura 2 parameter. So the parameter set is different, right? As I'm saying, like in case of Jukes Cantor, all the possible mutations will be explained by a single parameter. Okay. Whereas in case of Kimura 2 parameter, you will have one rate for transversion, another rate for transition. Okay. So transition and transversion are two types of mutations that we are talking about. Like we know that there are two different types of bases, right? Purine and pyrimidine. So purine to purine conversion versus purine to pyrimidine conversion, they are using two different rates for that. Okay. No. So the last model is just the most complex model. So as we are going down this table, the model complexity is increasing. Okay. More or less increasing. I would not say like continuously increasing all the time because sometimes they are having more flexibility in terms of one thing but not the other. But this last model is the most complex model, that it is the most generalized model. Okay. So again, this is problem dependent, that when you have your own data set, one thing that you can try, you can apply multiple of these models and then you can see under which model your data is explained better. And again, you can use something like AIC or BIC for that. Okay. Again, that depends really upon your data set. It varies from one problem to another. Generally, so again, when we are talking about length of a gene, let's say, okay, when we are looking at something like gene tree, megabases, it can be kilobases also. Maybe we are looking at only the exons of a gene. Yes, all of them are like equilibrium frequency is basically the same for all the bases. Mm-hmm. So that's what I mean. We are talking about a very computationally challenging problem, and you will see even the likelihood computation. It is quite expensive. Okay. Yeah, I think I have 15 minutes left, but I have not yet started with a single method. So maybe I'll just go there quickly. Okay. Um, Sure. I think we'll do that because otherwise it will be very difficult to finish. So let's actually talk about some of these distance-based methods. Okay. Again, I think we have already talked about all these things. So essentially, what we are trying to compute is this kind of evolutionary distance between any two sequences. So what is it? It is basically an estimate of the number of mutations that has occurred since those sequences diverged from their common ancestor. Okay, so this is the definition of evolutionary distance. Now, the one way that we can really compute this distance is we can compute the expected number of nucleotide substitutions that are happening for every site. And this is just an average measurement or expectation over the number of sites that we are talking about, right? And if we assume this, that if we assume that the evolutionary rate is constant over time, the distance is actually going to increase linearly with the time of divergence. Okay. So let's actually see a very simplistic measure of distance. So if we say that, okay, we have two sequences, okay, of length L, we are talking about ungapped alignment, so there is no gap, no indels. All we are taking care of is substitution kind of mutations. And D is basically the differences, the number of positions that are different in these two sequences. Then we can compute something like this, which is the ratio of D and L, and we are calling it something as P distance. So D is basically the number of positions at which the two sequences differ. L is basically the length of the sequences. Okay, we are assuming that the lengths are same. So this is known as P distance. This is very simple. Like we are saying, okay, 10% of the nucleotides differ, 20% of the nucleotides differ, something like that. Now, this is exactly the point that was raised earlier, that we are trying to model this kind of parallel or back mutations, because of which always the P distance is going to underestimate the number of substitutions that have really occurred in the data. Okay. So how do we see that through an example? So let's take a look at this particular example. We have an ancestral sequences. We have two different routes that have taken, and these are the two sequences in the end. Okay. So what we see here is that lot of substitutions are happening. T going to C, going to A, then C going to T, A going to G, going to C. So you can see same site being, I think, mutated multiple times during the history. If you count the actual number of total mutations that happened during this process, you will say you can actually count. But if you look at the P distance between these two sequences, you will see it is basically 2 out of 8. So 2 positions are different. But we can already see that so many mutations really happened over here, right? So there are 10 substitutions which happened during the evolutionary history, but we are just saying that 2 positions are different. So that means P distance is always underestimating the actual evolutionary distances, right? So what is the way for correcting this? So if we assume that essentially the probability of mutation at a particular site follows a Poisson distribution, then we can actually compute a corrected distance from this observed P distance. So how do you do it? Let's say we assume that there is a uniform mutation rate R, okay, per site, per unit time unit, per time unit. Then after time t, what is the average number of mutations? It will be RT, R multiplied by T, right? So the probability that n mutations have occurred at a given site during time t is basically given by this Poisson equation, right? Now, if there are two sequences that diverged really time t ago, the probability of neither sequence having mutated at a site is e to the power minus 2 RT. So that is basically you are setting n is equal to 0, right? So e to the power minus RT for one sequence, and for the second sequence, you multiply it, you get back e to the power minus 2 RT. Now, what we are saying is that P is the observed distance, or the P distance between these two sequences. The probability, and that means it can be thought of as the probability. P is really the probability that there are certain mutations. That means 1 minus P is really the probability that no mutations have occurred. So if that is the case, then 1 minus P, we can equate it to e to the power minus 2 RT. And we can say that this distance D, which is really the evolutionary distance, that is basically 2 RT, because RT is the average number of mutations in one sequence, right? So RT in one sequence, RT in the other sequence, we are getting 2 RT, And using this, we can now solve this equation to get to represent D in terms of P, right? So that will actually give us the corrected distance. That if you have P as the observed P distance, you can get the Poisson corrected distance, which is going to look like this. And then you will see that this is under the Poisson model, which is again a very simple model we are using for the mutations. There are gamma distribution that are also being used to make it a little bit more generalized. Now, again, these are very simplistic measures. Even we can talk about, let's say, the Jukes-Cantor model, which is the smallest or the simplest model of evolution that we just talked about, right? So let us see how the distances can be corrected if we are assuming that kind of models. Let's say Jukes-Cantor model. So it turns out that we have to somehow come up with this kind of probabilistic model which can describe the changes between nucleotides over evolutionary time, right? And again, going back to Shaon's talk where he was talking about discrete type Markov chains, in this scenario, since time is continuous, we are representing this kind of branch lengths in terms of expected number of mutations per site. So what is commonly used is something called continuous time Markov chain, right? Because we are looking at really these processes where we are dividing time into very short time intervals, and we are looking at the changes. So what are the assumptions? We are assuming that the nucleotide sites are evolving independently of each other. So this is a simplifying assumption. This we have to make, otherwise we'll just blow up the computation and will not get anywhere. Secondly, we are saying that substitutions at any particular site are described by a Markov chain, okay, where there are 4 states of that Markov chain, and the 4 states basically correspond to 4 nucleotides, A, C, T, and G. And apart from Markovian property, we also have some constraints on the substitution rates, okay? And this is exactly what is leading to different models of evolution. So let's actually see the Jukes-Cantor model. This is the simplest model. All we are saying that this is the rate matrix. Rate matrix is Q. Again, going back to Shaon's talk where he mentioned that you should not be confusing rate matrix with the transition probability matrix, okay? So this is what we are saying, that this is the rate matrix. The it's not probability, rate of change from one nucleotide to another nucleotide, that is one state of the Markov chain to another state of the Markov chain is parameterized by this alpha. And then you are seeing that as Shaon mentioned that the sum should be 0 in case of rate matrix. So that is what is happening here, that each row should sum to 0. So the diagonal elements becomes minus 3 alpha, okay? So this is very good. We have a rate matrix. How do we convert it to transition probability matrix, right? Because ultimately we will need that. We need to calculate the probability that given the nucleotide I at a particular position at a particular time, what is the probability that it will become nucleotide, some other nucleotide after time t, okay? So that is what is being modeled by this transition probability. So this theory of continuous time Markov chain is giving us this, that we can exponentiate this rate matrix, okay? Q is there and we are multiplying that with t, so that gives us this qt. We compute the matrix exponentiation of this qt using this series, and then we'll end up with another new matrix pt, which is again state cross state probability matrix, okay? And that is going to give us the transition probabilities, the different transition probabilities that we require. And if we apply this in this particular context, that is the Jukes-Cantor model, we are going to end up with 2 forms. So this is the probability of, that is the transition probability of I becoming j over a time t. Is this really the case? That is, it's remaining the same. It's not changing over time t. And the probability of changing it to some other nucleotide is this. So these are basically two entries, two possible entries in the transition probability matrix. Again, there are a lot of steps that I'm actually jumping over here. I'm just showing you the final result. So one other thing to remember is that we are always going to estimate alpha and t together. Just by looking at this or just by working with this, it's very difficult to distinguish alpha and t, okay? Because all we are having is the sequence data. So alpha t together becomes a single parameter of the model. Once we have this, we can try to solve the issue of, let's say, correcting the distance, okay? And then we can solve this particular equation, again, that is coming from the previous transition probabilities. Essentially, we should expect that there will be 3 alpha t mutations during a time t for each sequence site on each sequence. And after doing some more maths, what we'll end up with this corrected distance under the Jukes-Cantor model, which will be looking something like this, where p is basically our observed distance, that is the p distance, okay? Again, I think, jumped a lot of steps over here, not going into the details just for the sake of time, all right? But I just wanted to show you how this theory of continuous time Markov chain really is used in defining these models of evolution. Now, this is the simplest model. Again, if you change the Q matrix by introducing more and more parameters, you get back more and more complex models. Okay? So now coming back to this question of distance-based phylogeny problem, you are starting with a set of sequences. Essentially, you are given this kind of pairwise distances that you have computed by first creating this p distance and then converting it to some corrected distance by using some of these parametric models. Let's say you have a pairwise distance matrix that is the input to your distance-based phylogeny algorithm. The output is basically a tree structure T where the leaves are labeled with different sequences, and then consistent with M, where M is basically the model of evolution. Okay, so I'm going to talk about this particular method, which is UPGMA. Okay, what is UPGMA? The full form is unweighted pair group method using arithmetic averages, and it has certain assumption. It is assuming this molecular clock property, okay, and as a consequence, it is inferring a rooted ultrametric trees. The method is very, very simple, okay. What is the main idea? The main idea is that the two sequences with the shortest evolutionary distance between them, they are assumed to have been the last to diverge, okay. So if you consider, basically consider every pair of sequences, you can always find the pair of sequences with the least evolutionary distance, and they are the last to diverge, okay, and therefore they must have arisen from the most recent internal node in the tree. So this is the main idea. Yes. Yes. No, Jukes-Cantor is it's actually talking about something else. So Jukes-Cantor is basically when you are looking at a particular branch, let's say, you can apply Jukes-Cantor or whatever. Now, what UPGMA is saying that not just that, but across different branches also you have to assume the same parameter. Okay. So now let's come to the algorithm itself. So again, there is this initialization step where we are saying that each sequence is assigned its own cluster. Okay. So essentially, you start with the scenario that if you have 10 sequences, you will assign 10 of them to 10 clusters, each sequence to its own cluster, which is fine. Then This is the iterative approach. You have to find two clusters X and Y whose distance is the smallest. Okay, so that means you are going to take a look at all the pairwise distances in the distance matrix. You are going to choose a pair of sequences for which the distance is the minimum. Okay. The distance matrix is already given to you, that you have already computed. Okay. So it's just a matrix. Let's say you have 10 sequences. It's a 10 by 10 matrix. And you are just looking at the matrix. You are finding which entry is the smallest. That is already corresponding to two sequences, right? One on the column, one on the row. So if there are several equidistant pairs, you can pick one randomly. Fine. Now what we are going to do, we are going to create a new cluster XY, which is nothing but the union of the two clusters X and Y, and add it to the set of the clusters, and the individual clusters are removed. X and Y are removed. Instead, we add a new cluster in the set of clusters. Okay, so that is what we remove the other two clusters, and then we compute a new distance between XY and every other cluster in the set. How do we do that? I'm going to show you in the next slide. And then we repeat this process until only one cluster is left. So every iteration of this process is reducing the number of clusters. So at some point we are going to left with just one cluster, which is the whole tree, right? So that's when we stop this algorithm. So now the question is, when we are merging two clusters, then we are also going to compute this kind of distances, right? So the evolutionary distance between two clusters X and Y, how are we going to define it? It is nothing but the arithmetic average of the distances between their constituent sequences. So if nx is the number of sequences in X, cluster X, and ny is the number of sequences in the cluster Y, this is how we are going to compute the distances between after merging, right? This is the new cluster that we are talking about. And after we have created this new cluster, we have to also compute its distance to every other cluster, right? So how do you do that? Again, simple arithmetic average, weighted average. Nx multiplies by dxw plus ny dyw divided by the total number of sequences. So all very simple mathematical, I would say, formulations where we are just computing averages. Dijs are basically the entries in your distance matrix.
Those were coming from these Markov chain.
Yes, whatever you have done earlier. Yeah. So that's all. I mean, now let us take an example so that it becomes even easier. So this is what we are starting with. Let's say we have 6 sequences, A, B, C, D, E, F. Okay. Yeah. So the mutations, the data of the mutations is already being converted to, the distances. So, no. So the mutations, the information about the mutations are already being incorporated in the calculation of the distances. Before that already, because you were starting with the alignment data, you are looking at what positions are different, whichever positions are different, they are the result of a mutation, right? So you are converting that into first observed p distance, then you are applying this kind of any evolutionary model that you want to compute the corrected distance, and that corrected distance basically becomes your input. Okay. So in this particular matrix, what you see is that the distance between A and D is the smallest. So that's what we are going to choose first. We will say that A and D are the closest to each other, so they are the last one to diverge. So that means we have to form a new cluster using A and D. Okay. So that is what we are going to do, that we are going to create a new cluster V, which is A and D combined. So this is the V. Okay. And after we have done that, that means we have to compute the distance between V and all these other sequences. A and D are going away. So we no longer have, in the distance matrix, we no longer have A and D, okay? They have been removed, instead we have V. And these distances, sorry, have been again calculated using the formula that I have shown you. So with this new distance matrix, what we see, E and V are the closest to each other, right? So we combine them into another cluster, W. So we connect them through these branches, and we know basically, again, we use the previous formula to find out the distance between these two sequences or these two clusters, right? So we repeat this process, and after repeating this process, we'll end up with, each time we are basically merging two nodes from the distance matrix. And by repeating this process, then finally we are ending up with one entry in the distance matrix that is between the last two sequences to basically combine. This gives us the complete tree. So what you see here is that this ultrametric property is also ensuring that from the root to the leaf, the distance that we traverse is going to be the same, whichever path we take. Okay? So this is the distance-based method, UPGMA, which is again the very simple method. But in practice, people actually tend to use neighbor joining a little more, I would say, because neighbor joining is, first of all, there is a more generalized property that it is using. It is not assuming that same constant rate of evolution, okay, over time. So from ultrametric tree, it is basically getting rid of this molecular clock property. It is going to infer an additive tree instead. And it is also relying on a particular model of evolution where it is talking about minimum evolution, that the tree, the true tree will be that one for which the total branch length is the shortest. So we are actually looking for a more parsimonious model. We are saying that out of all the different possible evolutionary histories, whichever is going to give us the simplest evolutionary history is the more plausible model. Again, I am not going into the details of this method because that would be probably another 30 minutes. But if you are interested, I can share you the actual paper or some other reading materials. So now I come to the sequence-based methods. Here, initially, I plan to do, I think, talk about two different methods, but I'll jump onto just the likelihood-based methods without going into the parsimony-based methods. Okay, so what we are going to describe very quickly, because then it will be helpful for the next talk, we are going to describe a maximum likelihood approach of building this kind of phylogenetic trees. So what is likelihood? I'm going to simply define likelihood. Let's say you have a model M and you have a data represented as D. The likelihood of the model given the data is nothing but the probability of data under the model. Okay, so this is the simplest definition of likelihood function. You have a model, you have a data. The likelihood of the model is nothing but the probability of the data given the model. So let's take a very simple example. Let's say we have this data set where you are tossing 10 different coins. This is the outcome. Head followed by 4 tails, another head followed by 4 tails. Let's say you have 3 different models, model M1, M2, M3. M1 is assuming that you have a fair coin, head and tail are equally probable. M2 is saying that it's not a fair coin, it's biased coin, by the way. M2 is saying that probability of head is 0.8, probability of tail is 0.2, and M3 is saying probability of head is 0.1, probability of tail is 0.9. So what you do, again, there is no Markov chain here. All the outcomes are independent of each other. What you calculate the likelihood function, likelihood of M1, M2, M3 for the given data, that is this. And you basically, that is just the probability of the observed data, right? So in the first case, this is the probability. In the second case, this is the probability, and this is the third case probability. And you will see that under M3, this probability is the highest. That is, the likelihood is highest for the model M3. Okay. So in this case, then we are going to say that out of all the three models, this is the maximum likelihood estimate. M3 is the most likely model to explain the data. So essentially, our problem of interest is to infer the model M from the observed data, and this maximum likelihood estimate is basically going to be represented by this equation. M hat is the maximum likelihood estimate, where we are basically iterating over different models M, and we are computing the probability of the data given the model. We are going to choose the model with the maximum probability, okay, maximum likelihood. That is how maximum likelihood estimate is defined. Now, in case of phylogenetic tree, what is the data? We have a multiple sequence alignment as the data, right? And then what is the model? We are considering a model to be consisting of three things: the model topology T, the branch lengths, and the model of evolution, either Jukes Cantor or whatever. Let's say it is denoted by E, okay? So all these things are comprising of the model. Now usually then we are going to define the maximum likelihood estimate as something like this, that ultimately our solution should be consisting of the tree topology, the branch lengths, and the model of evolution. In practice, what is done, the model of evolution is first inferred from the data, okay? Again, using the techniques that I just described earlier. And then phylogenetic inference is, during the phylogenetic inference, we are already going to assume that the model of evolution is fixed. We are not going to change it, okay? So E hat has already been determined. So essentially, we are left with two things, T and lambda. These are the things that we have to determine. That is the phylogenetic tree topology and the branch lengths associated with the phylogeny. So how do we do it? So this is our input, a matrix of the taxa characters. Again, this is our input data. This is our input data. Let's say, I don't know, let's say there are m columns, right? And n different taxa, okay? So this is our input data, alignment data, and the output is basically a tree which is denoted as T, where the leaves are labeled by this set of taxa, n taxa, and then we are also going to have the branch lengths such that this likelihood is maximized. The probability of the data given T comma lambda, this is maximized. So now one simplifying assumption is that we are going to make is that we are going to say that each site is basically independent of each other, okay? So that means here, okay, here n is denoting the number of sites. Let's change this notation a little bit, okay? Each row is a sequence, yes, okay? So let's say that there are n sites in the sequence, and then m is basically the number of sequences. Again, the transpose of this matrix is drawn over here, really, okay? Don't get confused by that. So this is basically saying that there are n sites in the alignment, m total sequences. So the overall likelihood function is going to be nothing but the product over the number of sites, and for each site we have to compute this probability, okay, which is the probability of dj given T comma lambda. And then it is basically going to be something like this, where R is basically representing the root. So we'll come to that, okay? So this quantity, again, we are repeating this for every possible sites, but that means somehow this needs to be calculated very efficiently as well, Okay, and this will just give us the likelihood of one particular tree. So what we need to do iteratively is that we have to start at a particular tree topology and let's say a set of branch lengths. We evaluate the likelihood and then we stochastically move to other branch lengths or other tree topologies in order to explore the overall space of possible trees. And there are existing algorithms for doing that. There are hill climbing or stochastic search methods which can be used. All I'm going to show you is how do we calculate this likelihood, okay? So let's say.
So is it understood to what extent the landscape is smooth or rough or anything like that? Just people just do it and.
People just do it, and what is usually works in practice is that you will run it multiple times, and
Just
the
start at different places.
yes,
So
do random
just do it
restarts.
until you're happy.
Yeah. So because that's the best you can get to.
That is like a humongous number of trees.
Yes, yes, and usually it is preferable that if you return multiple trees rather than just working with a single tree. So that's why Bayesian methods gained more popularity later on. That you are actually having multiple solutions and you can see how many times a particular branch is appearing in your trees. Okay, so I'll just quickly talk about this likelihood computation because this is a very interesting dynamic programming algorithm which is applied on the tree structure itself. Okay, so let's say this is our tree. We have a root denoted as R. These are the set of leaves and these are the internal nodes. So this is a candidate tree structure for which we are trying to compute the likelihood. Okay, so let us define this particular quantity Cj of x comma v, where j is basically denoting the site, right? And x and v are basically saying that this is the probability of a subtree where the root is denoted by the node v. Let's say this is an internal node, right? Such that the character or the residue at the jth position of v is x. So this is something like a partial likelihood where x can take any value, a, c, t or g, right? According to our problem definition. Then we'll start from the bottom itself, that is at the leaf level. So leaf level, the data is observed. It's very simple. Whatever data is observed at this particular scenario, for that the partial likelihood is 1. So let's say if Vj is a, then Vj of a or Cj of v comma a is going to be 1, otherwise it is going to be 0, right? Because this is our observed data. What is most interesting is when we are looking at any internal node. So what we are doing over here, what you see is that V is an internal node with two children, U and W, okay? So because we are doing it bottom up, we are constructing this partial likelihood from the leaf towards the root. So what will happen? That means when we are computing this partial likelihood for V, U and W have already been considered, right? We already have their partial likelihoods. So what you are seeing over here is that this is basically product of two different terms. This is coming from U and this is coming from W. What is this? This is basically saying we are marginalizing over all possible mutational histories. Okay, so that is what we are considering. Over here you see there is a summation over y. Y is nothing but these values a, c, t and g, because we are computing these partial likelihoods, right, for a particular node and particular character. So we are doing that over here, that this is the partial likelihood for u, the node u, and this y. Y is basically any value within a, c, t and g, and that should be multiplied with a transition probability, that probability of x to y, that is there is this particular node for which we are saying the character is x, and this is the node for which we are saying the character is y. That means there is a nucleotide change happening from x to y. Now x and y can be same, so that also has to be accounted for, but that is happening over this branch which is connecting u and v. Okay, so this is the branch connecting u and v, so this is the branch length. which is giving us the transition probability. And this is the overall contribution of this particular node over here, U. Similarly, we have contributions from the other child, W. So this is how we can actually calculate the partial likelihood at the internal node V. Okay. So this is being repeated for every internal node and every possible character in X. Okay. And we move from the leaves towards the root. So in the end, acha, by the way, these transition probabilities are actually coming from the model of evolution. Okay. Because any model of evolution you take, that can give us these transition probabilities. This is for one possible transition event, and because you are taking a summation, that means you are marginalizing over all possible transition events. Okay. So finally, at the root, you are going to come up with all these different probabilities, right? So that means at the root we have all possible values of x, a, c, t, and g, and then we have all the partial likelihoods. What you are going to do, you are going to multiply that with p of x, which is basically the base equilibrium frequencies for AC, observing A, C, T, and G. Okay, and then if you are again marginalizing over those possibilities, you are then getting this overall likelihood of this whole tree structure. So this is exactly the algorithm which is known as Felsenstein's likelihood formulation. So this is essentially a dynamic programming algorithm on the tree. Okay. So I think I should stop here now, and of course we are already over time, 20 minutes. So maybe we can take a break and then we'll talk about cancer, like how these things are applied in case of cancer.